Okay, guys. This is a video podcast dealing with the material on transforming bacteria, and also it's a general introduction to DNA and our ability to generate what we call GMOs, or genetically modified organisms, through a variety of methods. And so, what we're going to talk about first off is the structure of DNA, how it's organized, how the information in DNA is used to direct the activities, the physical characteristics, and the physiology of the organisms in which it's found. So we'll address what we call the central dogma biology. And then we're going to move into some different systems of gene expression so that we can understand a little bit more about the physiology of gene expression, the molecular biology of gene expression. In other words, when is a gene used and when is it not? And what are the control mechanisms like in both prokaryotes and eukaryotes? And then we'll finish up with a discussion of some model organisms that we use in the lab to produce essentially transgenic organisms that we can study and as a result learn the roles of different genes in those organisms and use that information to basically understand a little bit more about other organisms, including ourselves. So we begin with a discussion of the structure of DNA. So take a listen.
The structure of the DNA molecule was first inferred by James Watson and Francis Crick, based primarily on X-ray crystallography data collected by Maurice Wilkins and Rosalind Franklin, and chemical analysis of base composition of DNA conducted by Irwin Chargaff. The key features of the structure are its right-handed double helical structure. Each helix consists of an alternating sugar-phosphate backbone with nitrogen bases projecting toward the interior of each helix. One complete 360-degree turn of the helix covers 10 bases of length and equals 3.4 nanometers in physical distance along the axis of the molecule. The width of the double helix is 2 nanometers. The nucleotide bases are attached inside each backbone of the molecule so that the nucleotides in one helix or strand are hydrogen bonded to the bases in the other helix or strand. The hydrogen bonds hold the two strands of the double helix together. An adenine in one strand is always paired with a thymine in the other strand, and a guanine in one strand is always paired with a cytosine in the other strand. Note that guanine-cytosine base pairs form 3 hydrogen bonds, while adenine-thymine base pairs form 2 hydrogen bonds. This makes guanine-cytosine base pairs more stable than adenine-thymine base pairs. Nucleotide pairing between strands also allows the sequence in one strand to determine the sequence in the complementary strand. The two ends of a strand are not identical. Note that on one end of each strand, a 3' hydroxyl group of the deoxyribose sugar is not involved in the backbone, or we say it's free, while at the other end of the same strand, the 5' hydroxyl group of the deoxyribose sugar at the end is free or may contain a phosphate that is free and not bonded to another deoxyribose sugar. This dissimilarity of the two ends of a strand creates the ability to uniquely distinguish each end of the strand. Because of this polarity of each strand, we can see that the two strands of DNA are oriented in opposite directions, or we say that they are antiparallel. In three-dimensional space, the two helices are not equally spaced along the axis of the molecule, such that a major groove and a minor groove are created by the asymmetric spacing of the antiparallel helix backbones. The major groove and minor groove play important roles in the binding of proteins that regulate gene transcription.
Now, as you noted in the video, DNA is a double helix with 2 antiparallel strands, and it obeys the rule of complementarity, where an A on one strand will always pair with a T on the other, and a G on one strand will pair with a C on the opposite strand. Remember, again, from the video that the strands run in opposite directions. And as a molecule, DNA has the ability to either assume its normal conformation, the double helical form that we were looking at just a minute ago, or it can unzip, unwind, in order to allow either for DNA replication to take place, that would be the production of new DNA molecules, or for transcription to operate in certain regions of the DNA so that we can carry that information that's in the nucleus out to the cytoplasm, where eventually we can turn that information, in most cases, into a protein molecule which will perform some task in the cell or in the organism. As a result, we have to appreciate that DNA can unzip or anneal depending on differences in temperature, in pH, and in ionic strength. So there's a way to compute the melting temperature of a DNA double helix, and it's given here, where the temperature of melting is computed as 81.5 plus 16.6 times the log to the base 10 of the molar sodium ion concentration plus 0.41 times the percent of GCs in that region of DNA minus 600 divided by the length. And what you see here as a result of this formula is that the higher the GC content, the higher the melting temperature, and the reason for that is that in GC base pairing there's 3 hydrogen bonds, whereas in AT base pairing there's only 2. So it's easier to melt stuff that has a lot of ATs in it than with a lot of GCs in it. And the higher the sodium content, the more difficult it is to melt the DNA double helix. And that's going to be a factor when we make up DNA solutions a little bit later on. We want to stay within a reasonable range so that the double helix doesn't unwind. If the double helix unwinds, it gets very difficult to manipulate DNA in order to do things like introduce it into other organisms, or to cut it at certain regions, or to ligate it together. So we have to be aware of this phenomenon, this melting temperature. Now, in addition to that, there's a major pH effect as well. DNA will denature at a pH of greater than 11.3 regardless of temperature. It'll also denature at a pH of less than 3. However, this will also depurinate and fragment the DNA molecule and make it essentially useless in terms of expressing genetic information, although it might prove useful in certain other techniques such as southern transfer. DNA comparatively is a pretty stable molecule. When you look at protein, DNA, and RNA, DNA is probably the most stable of the 3, but that doesn't mean that you can't ruin DNA. One of the things that we have to appreciate is that we're surrounded by an unsterile universe, right? The world is not pure and clean, and as a result, there are little microbes everywhere around us that secrete enzymes that degrade DNA. These are called DNases. In order for these DNases to work, they require a certain pH, and they also require magnesium as a cofactor. And so when we make DNA solutions, one of the things we're going to take special care to do is to control the pH, number 1, control the temperature, number 2, and also to take special care to get any magnesium out of solution that we can, so that those nucleases can't wreak havoc on our prep, okay? Because we don't want our DNA to break down. By comparison, RNA is quite fragile. RNA differs from DNA in 2 respects. Number 1, it's generally single-stranded. Number 2, instead of thymidine as a nucleotide, it uses uracil. And more importantly, it doesn't use deoxyribose as the sugar in the sugar phosphate backbone. Instead, it uses ribose. And ribose, as a result of having this extra hydroxyl group at the 2' end, can potentially form a phosphodiester bond where we can actually cleave the RNA molecule on its backbone as a result of this intramolecular hydrolysis reaction. So to avoid RNA breakdown, this inherently unstable molecule has to be handled with buffers treated with DEPC, which are then autoclaved. DEPC stands for diethyl pyrocarbonate. This also applies to plasticware and glassware, which might be used to work with RNA. The DEPC essentially adds carbamyl groups to the active sites of RNA degrading enzymes and renders them inactive. We're not going to work with RNA in this course. It takes a separate facility, and it takes some fastidious preparation in order to effectively work with RNA. You may work with RNA down the road in a lab setting, and what you'll find generally is that in labs that deal with RNA, you'll find that those facilities are separate and much cleaner than the areas where we work with DNA. Take note that RNases and other nucleases are ubiquitous, so always assume that your materials are contaminated with these enzymes unless they've been treated. Okay, so that's the difference between the two molecules chemically. Now, what we're going to learn here in a minute when we look at the central dogma of biology is that the flow of information in living cells goes from the nucleus in blocks of information that are called genes that are contained in the DNA. It's then transferred in a process called transcription to molecules of messenger RNA, and then those messenger RNA molecules are used to direct the synthesis of protein in a process that's called translation. Now, the basic setup is the same in prokaryotes like bacteria and eukaryotes like plants, or you or me, or a mouse, for instance. But there are some slight differences. One of the things that we have to appreciate about the central dogma is that in eukaryotes, the transcription and the translation happen in physically different compartments, right? The transcription happens in the membrane-bound nucleus, while the translation occurs in the cytoplasm. So that's one difference. The other difference is that in prokaryotes, transcription and translation happen simultaneously often, where the transcript, which is made straight off the DNA molecule, is immediately bound by ribosomes and used to direct the synthesis of proteins. And as a result, translational rate and transcriptional rate often have a big influence on gene expression. So let's take a look at the central dogma here, and I'll join you in a minute. Okay, so notice that DNA is remarkably adapted as a storage molecule.
The DNA double helix contains two linear sequences of the letters A, C, G, and T, which carry coded instructions. Transcription of DNA begins with a bundle of factors assembling at the start of a gene to read off the information that will be needed to make a protein. The blue molecule is unzipping the double helix and copying one of the two strands. The yellow chain sneaking out at the top is a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA letter by letter to copy the gene. At this point, the RNA needs to be edited before it can be translated into a protein. This editing process is called splicing, which involves removing the green non-coding regions called introns, leaving only the yellow protein-coding exons. Splicing begins with assembly of factors at the intron-exon borders, which act as beacons to guide small proteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the two exons together. The edited RNA and intron are released, and the spliceosome disassembles. This process is repeated for every intron in the RNA. Numerous spliceosomes remove all introns so that the edited RNA contains only exons, which are the complete instructions for the protein. Again, this is happening in real time. When the RNA copy is complete, it snakes out into the outer part of the cell. Then all the components of a molecular factory called a ribosome lock together around the RNA. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. Inside the ribosome, the RNA is pulled through like a tape. There are different transfer molecules for each of the 20 amino acids shown as small red tips. The code for each amino acid is read off the RNA 3 letters at a time and matched to 3 corresponding letters on the transfer molecules. The amino acid is added to the growing protein chain, and after a few seconds the protein starts to emerge from the ribosome. Ribosomes can make many proteins. It just depends what genetic message you feed into the RNA.
The reason for this is that the instructions that direct the synthesis of the second strand of DNA are contained in the opposite strand of DNA, as the enzymes use that as a template to direct its synthesis. This is important because this helps to ensure the fidelity of genetic information as it's passed from organism to organism. Okay? It wasn't until the elucidation of the double helical nature of DNA that we figured out how it served as an effective way to transfer information from cell to cell and from organism to organism. It's this complementarity and the fact that each strand contains information to direct the synthesis of the opposite strand that allows it this unique ability. Okay, this is just a look at simple gene expression.
The sequence of nucleotide bases in DNA carries genetic information in units that are called genes. Structural genes encode the information for specific proteins. To create a protein, a gene must first be transcribed into a sequence of nucleotide bases in a messenger RNA molecule. The messenger RNA sequence is then translated into an amino acid sequence of a protein. This sequence of amino acids in a protein molecule determines the shape and chemical characteristics of the protein. Thus, each structural gene specifies a specific protein in the cell that carries out a specific function based on its chemical characteristics and molecular shape. This function of the specific protein gives the cell and the organism the specific trait coded by the gene.
Okay, next thing we'd like to do is to compare gene expression in eukaryotes with gene expression in prokaryotes. So take a listen here.
Gene information is processed differently in prokaryotes and eukaryotes. Because prokaryotes lack a nuclear envelope, messenger RNA, mRNA, can associate with ribosomes in the cytoplasm as the mRNA is being formed. Translation of mRNA into protein begins before transcription is complete. Individual bacterial mRNA molecules often contain transcripts of several genes. By placing genes with related functions on the same mRNA, bacteria coordinate the synthesis of these proteins. These clustered genes are referred to as an operon. Because eukaryotic cells possess a nucleus, their mRNA must be completely formed and must pass across the nuclear envelope before translation. Eukaryotic mRNAs are modified before they are translated. Introns are removed and the remaining exons spliced together. A 5' cap and a 3' poly A tail are added. The processed mRNA travels to the cytoplasm where translation occurs. In contrast to prokaryotic mRNA, eukaryotic mRNA usually specifies only a single protein.
So one of the things that you've noticed about these two systems is that they're both cells. They both utilize DNA as genetic material. But in prokaryotes, transcription and translation happen in the same compartment. Also notice in prokaryotes that the genes generally occur as uninterrupted blocks, so there is no information in the DNA that does not show up in the messenger RNA. Also note that often transcription and translation are coupled in prokaryotes. In addition, notice in eukaryotes that transcription and translation occur in different compartments. Also note that the messenger RNA is processed. It is capped, spliced, and polyadenylated before it's escorted into the nucleus, where translation will occur either in the cytoplasm or bound to the rough endoplasmic reticulum. It was long thought that these gaps in DNA information that don't show up in eukaryotic messenger RNA were essentially junk DNA, that they served no purpose. Turns out that nothing could be further from the truth. We've learned relatively recently that these introns contain pieces of information that are used to direct the synthesis of RNAs that don't necessarily code for a protein, but can direct the expression of the gene in which they're found, as well as other genes, by either knocking down or enhancing the expression of the nascent transcription product. We've also learned that these spacer regions, these introns, contain critical sequences that direct the splicing and the processing of messenger RNA. We've found a variety of genes in eukaryotes that are alternately spliced and as a result generate a variety of different gene products from a single gene. In prokaryotes, we find something rather interesting, which is that frequently we'll see genes that may overlap and may be transcribed in opposite directions from each other, as a result making very efficient use of the genome. Again, no introns in prokaryotic DNA, but because the genome is compact compared to the eukaryotes, we try and make as much use of the genetic space as we possibly have. Now, also notice that in eukaryotes, another major difference is that we generally have more than one chromosome. A chromosome is simply a DNA molecule. In eukaryotes, that genetic material is wound around special proteins that are called histones that serve to compress and compact the genetic material, both to avoid damage as a result of the open chromatin configuration, but it also turns out that these histones play a role in gene expression, and this is something, again, that's been learned relatively recently. Histones, remember, are absent in prokaryotes as well. Notice also that the capping and the polyadenylating play a role in eukaryotic transcription. The capping of the 5' methylguanosine cap at the 5' end of the message serves to direct the message out of the nucleus and into the cytoplasm. It also serves as a starting point for translation, something essentially to latch onto as the ribosomes bind and scan for the appropriate reading frame within that message. Also note that the poly A tail is thought to direct the lifetime of that particular message. The longer the tail, the longer the message stays around. And it has been relatively recent that we've discovered that the messenger RNA in eukaryotes is not in a linear form when the cytoplasm, when it's translated, it actually circularizes in many cases, and as a result, the ribosomes march essentially in a circuitous route from the 5' methylguanosine cap all the way to the end of the message. Another important thing to note is that the ribosomes in eukaryotes and prokaryotes, while functionally similar, are biochemically different. They do the same job, but if we were to break them down and look at them in terms of their protein and RNA components, there would be significant differences. It's also notable that the translation and transcribing apparatus found in eukaryotic mitochondria and chloroplasts strongly resembles that of prokaryotes, particularly the bacteria. So the thinking currently is that chloroplasts and mitochondria were obligate endosymbionts that resulted early in evolution in a very beneficial relationship between the earliest eukaryotic and prokaryotic cells. The chloroplastic endosymbionts providing photosynthetic apparatus for the early plant cells, and at the same time the mitochondria providing an energy powerhouse for the early eukaryotic cells. The eukaryotic cell, of course, providing shelter and a source of food for these, what eventually became eukaryotic organelles. Okay. All right, so we want to take a look here at prokaryotic gene expression as exhibited by some inducible and some repressible operons. So let's take a listen to prokaryotic gene expression. Most of this work is done in E. coli.
Many bacteria grow on glucose before they utilize other compounds, such as lactose, as a growth substrate when both are present in the medium. Transcription of the lactose operon is controlled by two regulatory proteins. The catabolite activator protein can bind to the activator binding site and facilitate transcription, and the lac repressor protein can bind to the operator site and block transcription. The catabolite activator protein can bind to the activator binding site only if cyclic AMP is bound to it. When glucose is present, the concentration of cyclic AMP in the cell is low. RNA polymerase cannot bind efficiently to the promoter, and transcription does not occur unless the catabolite binding protein is bound to the activator binding site. Therefore, transcription of the lac operon does not occur in the presence of glucose. In addition, if there is no lactose in the medium, the lac repressor binds to the operator site, and this blocks transcription. When both glucose and lactose are present, there is insufficient cyclic AMP to bind to the catabolite activator protein, which therefore cannot bind to the activator binding site. Without the bound activator protein, RNA polymerase cannot bind to the promoter and initiate transcription, even though lactose binds to the repressor and prevents it from binding to the operator site. When neither glucose nor lactose is present, the concentration of cyclic AMP is high, and the catabolite activator protein is bound to the activator binding site. RNA polymerase can bind to the promoter, but it is blocked by the repressor on the operator site. When glucose is absent and lactose is present in the medium, cyclic AMP is present and binds to the catabolite activator protein, which in turn binds to the activator binding site and facilitates transcription. Lactose binds to the repressor and prevents it from binding to the operator site. RNA polymerase can now bind to the promoter and carry out transcription.
Okay, and here's a look at regulatory proteins and how they affect gene expression in prokaryotes. We're looking here at the arabinose operon.
When a microorganism such as E. coli is grown on glucose, it must synthesize all of its amino acids. If an amino acid, such as histidine, is added to the medium, the organism can turn off the synthesis of the enzymes needed to make histidine. In the absence of the amino acid, a regulatory protein called a repressor is made, but it is unable to bind to DNA and prevent transcription. When the amino acid is added to the medium, it enters the cell, reacts with the repressor, and activates it so that the repressor now binds to the operator site on the DNA and turns off the pathway by blocking RNA polymerase and preventing messenger RNA synthesis. When microorganisms such as E. coli are grown on glucose, they do not synthesize the enzymes needed to degrade other growth substrates such as lactose. In the absence of lactose, the regulatory protein, called a repressor, binds to the operator site and blocks transcription. When the substrate, which is called the inducer, becomes available, it reacts with the repressor and inactivates it. The repressor can no longer bind to the operator site and transcription can proceed. Regulation of catabolic pathways involving a repressor that is inactivated by the inducer is referred to as negative control. Some degradative pathways are regulated by an activator protein. An activator is a regulatory protein that facilitates transcription by binding to DNA. Activators must react with the substrate that they regulate, such as arabinose, before they can bind to the DNA. The binding of the activator to the activator binding site on DNA enhances the ability of RNA polymerase to bind to the promoter site. In the absence of the inducer, the activator cannot bind to the DNA. RNA polymerase is unable to bind to the promoter site and transcription does not occur. Regulation of degradative pathways involving an activator that is activated by an inducer is referred to as positive control.
And here we see a repressible operon. This is the tryptophan locus, again characterized in E. coli. So take a listen.
In the bacterium Escherichia coli, a group of 5 genes code for enzymes required to synthesize the amino acid tryptophan. All 5 genes are transcribed together as a unit called an operon. An operon is a group of genes that is under the control of a single operator site. A regulatory protein called a repressor can bind to the operator site and prevent transcription. When tryptophan is lacking in the environment, the repressor is made, but it is unable to bind to the DNA and block transcription. RNA polymerase binds to the promoter site and then proceeds down the DNA, transcribing the genes for the tryptophan biosynthesis enzymes. When tryptophan is present in the environment, the organism no longer needs to make tryptophan. Tryptophan binds to the repressor and activates it. The activated repressor now binds to the operator located within the tryptophan promoter and blocks transcription.
Okay, so what you've noticed about prokaryotic gene expression is that it's tailored to the needs of the cell. Essentially, the idea is that we want to produce the gene product only when it's needed in order to meet the metabolic needs of the cell in the case of the lac operon, the tryptophan locus, and the arabinose locus. And this only makes sense because we don't want to waste cellular resources, machinery, amino acids, ATP, what have you, performing an activity that's not going to provide an advantage for the cell. So you can see that there are a variety of methods of control. We have the inducible operon, we have the repressible operon. Basically, the idea is that you have a gatekeeping function at the beginning of the gene that will either permit or not permit the transcription of that particular gene product, and that gatekeeping function is going to be either in the on or the off position based on the need for that particular end product of that enzymatic pathway. Notice also that transcription and translation are coupled in all these processes. As a result, it's incumbent upon translation activity that it proceed unimpeded regardless of secondary structure in bacterial message. And that can change depending on, again, the metabolic state of the cell at that time. Okay, so this is another level of control where essentially the control at the translational level is dictated by the ability of the transcription apparatus to generate a message that can be read from front to back. Now in eukaryotes we have a completely different situation. Okay, we have partitioning of transcription and translation in two separate compartments. The DNA is arranged in information blocks called genes. In these genes there are going to be areas of genetic material called introns that don't show up in the eukaryotic message in the final form, but that doesn't mean that these pieces of DNA are not important in regulating the activity of the gene. Once the nascent message has been produced, it's going to be capped, spliced, and polyadenylated and escorted out of the nucleus through nuclear pore, where it'll be bound to either by free or by membrane-bound ribosomes, which are resident on the rough endoplasmic reticulum, and then that protein product, once produced, in the case of membrane-bound ribosomes, will be extruded into the rough ER, where it will eventually find its way to the Golgi, and from the Golgi it will be loaded into a vesicle, which will eventually be sent to a final destination in the cell, where that protein will be dropped off and it will perform its function. In the case of cytoplasmic ribosomes, the message will be translated essentially in the cytoplasm and will immediately begin to perform its function. Remember that the lifetime of the message is in part dependent on the length of its poly A tail. Also realize that we can control gene expression at both the transcriptional and the translational level in the eukaryote. At the transcriptional level, we can control how much of the message is produced and how long the message exists in the cytoplasm, and this at the translational level will obviously have an effect on how much of the gene product is produced. Okay, so a couple of ways that we can modulate gene expression. The anatomy of the gene in a eukaryote is essentially, it's similar to the prokaryotic anatomy. There's a gatekeeping function at the 5' region called the promoter. There are going to be, in most eukaryotic genes, exons that represent the protein coding component of the gene, and introns, which we now know contain not only splicing elements, but also contain information that will direct the gene's expression, not necessarily just in that locus, but perhaps in other loci as well. And then there's also polyadenylation signals at the 3' end of the gene that direct the end of transcription and also direct where the poly A tail will be appended before the processed message ends up being escorted out of the nucleus. Okay.
Most of the control of gene expression in eukaryotes is achieved by regulating the frequency of transcription initiation. Various regulatory proteins bind to DNA and affect the ability of RNA polymerase to bind to the promoter site and carry out transcription. Efficient binding of the RNA polymerase increases the rate of transcription initiation. Gene expression can also be controlled by altering the splicing of exons in eukaryotes. For example, the faster messenger RNA is processed, the greater the amount of gene product. Gene expression can also be regulated by controlling access to or efficiency of transport channels. The passage of an mRNA transcript across the nuclear membrane is an active process requiring that the transcript be recognized by receptors lining the interior of the pores. Many enzymes degrade messenger RNA. Gene expression can be regulated by modulating the degree to which the messenger RNA is degraded. Many different proteins and amino acids take part in the translation process. The rate of gene expression can be regulated by speeding up or slowing down protein synthesis. The availability of any of the proteins or amino acids can alter the rate of the protein synthesis. Phosphorylation or other chemical modifications can alter the activity of a protein after it is produced. This is referred to as post-translational modification.
This is just a look at some of the 5' elements that can control eukaryotic gene expression. The idea here, again, is that we can determine the amount of message we produce by permitting or inhibiting the transcriptional apparatus from proceeding from the 5' to the 3' end of the gene. One of the mechanisms whereby we can enhance or increase the amount of transcription that occurs is through the interaction of the promoter with regions that are called enhancers. And what these enhancers can do is they can change the DNA conformation near the gene that they control. They can also serve as recruitment points for special proteins, which can promote, for instance, the assembly of a transcriptional complex, which will then in turn generate the message. Or we can also have, obviously, the reverse phenomenon, where we can inhibit the assembly of a transcriptional complex that would produce the message. Okay, and in this case, these would be repressors or inhibitors, and they can be found upstream of the promoter. They can be found within the introns. In some cases, they can even be found within the exons of the gene. Okay, so let's take a look at transcriptional complexes and enhancers.
For RNA polymerase to successfully bind to a eukaryotic promoter and initiate transcription, a set of proteins called transcription factors must first assemble on the promoter. The assembly process begins upstream from the transcription start site, where proteins called basal factors bind to a short TA-TA sequence in the promoter. Other basal factor proteins then bind, eventually forming a full transcription factor complex able to capture the RNA polymerase. Basal factors are essential for transcription, but cannot by themselves increase or decrease its rate. A second set of transcription factors, called coactivators, link the basal factors with a third set of transcription factor proteins called activators. Activators are regulatory proteins that bind to sequences on DNA called enhancers. Enhancers are located at sites that are distant from the promoter. The interaction of activator proteins with transcription factor subunits increases the rate of transcription. Many enhancers, scattered around the chromosome, can bind different activators, which provide a variety of responses to various signals. When a second kind of regulatory protein called a repressor binds to a silencer sequence located adjacent to or overlapping an enhancer sequence, the corresponding activator is no longer able to bind to the DNA.
Okay, now that we understand a little bit about how it is information flows in a living cell, from the nucleus to the cytoplasm via the message, and then from the cytoplasm to the protein via translation of the ribosomes of the message, we can now take a look at some of the different systems available to us and think about how can we explore this information that's contained in the nucleus in a way that we can understand what the genes do in the living system. Okay, and there's a variety of techniques that we can use to do that. We have the ability, for instance, to introduce foreign genes into a variety of organisms and then observe the change in the phenotype. We also have the ability to induce mutations in existing genes and observe changes in phenotype. We also have the ability to introduce novel genes from other systems or between systems and look at the effect on the phenotype as well. We can do this in bacteria quite easily through a variety of methods that we'll see in just a minute. And we can also do this in a variety of eukaryotic organisms. Now, I didn't touch on yeast in this particular talk because we didn't really do much with yeast in this particular installment of the course. But I did touch on plants in the form of Arabidopsis and animals, looking here at Drosophila and also at the common house mouse as systems that can provide information that's a little bit more applicable to the human condition. Okay, and so we start off our discussion with genetically modifying bacteria. So we're going to take a look here at bacterial transformation.
DNA transformation involves the transfer of naked DNA into a recipient cell. In the first step, double-stranded donor DNA binds to specific receptors on the surface of a competent cell. One strand of the donor DNA is degraded by nucleases while the other strand enters the cell. The single-stranded donor DNA pairs with an homologous region on the recipient DNA and is integrated into the recipient genome by a breakage and reunion mechanism called homologous recombination. If there are any differences between the nucleotide sequences of the donor and recipient DNAs, the mismatch repair system comes into play. The repair system removes either the donor or the recipient strand and replaces it with the complementary sequence. Since either strand may be repaired, some cells contain the new donor DNA and others have the original DNA sequences. In the laboratory, cells are plated on selective media so that only the transformants will grow.
Now, one of the things we have to appreciate about bacteria is that it's interesting in and of itself to study the genetics of the bacterial cell, but we should also appreciate the fact that the bacteria can also serve the scientist as a method whereby we can produce large quantities of genetic material to work with and to introduce into other systems, okay? Be they animal, be they plant, be they insect, whatever, okay? One of the biggest breakthroughs regarding this was the discovery and the construction of relaxed plasmids that have the ability to generate large numbers of DNA that's independent of the bacterial chromosome within the bacterial cell. These plasmids have several features that make them excellent shuttle vectors. They contain sites for proteins that are called restriction enzymes that can cleave the DNA in specific locations and allow us to insert DNA from other systems into these extra chromosomal pieces of genetic material. They contain selectable markers so that bacteria that take up the plasmid will be resistant to a drug, say like ampicillin or tetracycline or kanamycin, so that we can select only for those transformed cells that contain our construct of interest. And they often also contain a screenable marker where we can determine by looking, for instance, at a color reaction whether or not the bacteria not only have taken up the DNA that we've introduced, but whether or not that DNA contains a foreign gene inserted into a restriction enzyme sequence. Once we've done that, once we've isolated those bacterial colonies that satisfy those characteristics, we can then grow those clones up. We can isolate genetic material from those clones, and then we can manipulate it further and eventually introduce it into other systems, say a fly or a mouse or a plant or any of a variety of other systems that are being utilized today. So let's take a look at plasmid vectors.
Plasmids can be good cloning vectors because they carry an origin of replication and are therefore able to replicate independently within a cell. Most plasmids used as vectors also encode some type of selectable marker, such as the gene for resistance to ampicillin. If the host cells are ampicillin sensitive, the only host cells that can grow on a medium containing ampicillin are those that have taken up the plasmid. Vectors must also have a small sequence of base pairs that can be recognized by a restriction enzyme. When this enzyme opens the circular plasmid, foreign DNA can be incorporated. When the plasmid vector and foreign DNA are both cut with the same restriction enzyme and mixed together, not all molecules will join to form recombinants. Some vector molecules will reanneal without incorporating foreign DNA. To identify cells that contain plasmids that have incorporated foreign DNA, a second marker gene is needed on the vector. This second marker contains the restriction enzyme site within its nucleotide sequence. If foreign DNA is inserted, the second marker is inactivated. This is referred to as insertional inactivation. A common second marker is the LAC-Z gene, which codes for the enzyme beta-galactosidase. Beta-galactosidase can cleave a colorless chemical called XGAL to form a blue compound. Therefore, colonies of cells that harbor the intact vector but no new recombinant DNA can make beta-galactosidase and form a blue color in the presence of XGAL. However, colonies that contain new recombinant DNA cannot make beta-galactosidase and are white.
Okay, one of the other ways to modify bacteria is through conjugation. Conjugation is a process whereby two bacterial cells unite via the growth of a structure called a pilus, which is essentially a pipe that connects two bacterial cells together. The direction of the pilus, production of the pilus, is contained by genes that are on something called the F episome. The F episome can then transfer itself through the pilus from bacterial cell number 1 to bacterial cell number 2. The result being that both cells then can come away with a copy of the extra chromosomal DNA because the F episome can produce copies of itself through a process called rolling circle replication. Once the conjugation pipe is broken, the gene transfer stops. The interesting thing, of course, about this is that this is one of the ways that bacteria share drug resistance, not only within a bacterial subtype, but between bacterial subtypes. As a result, you can imagine that bacteria with resistance to different drugs can conjugate, swap genetic material, and then come away from the conjugation resistant to both forms of the drug. And this has resulted in the production of what we now today call superbugs that are resistant to a variety of antibiotics simultaneously. So let's take a look at conjugation.
Conjugation is a mechanism of gene transfer that requires direct contact between donor and recipient cells. A plasmid is a small piece of DNA, separate from the main chromosome, that carries genetic information for such things as antibiotic resistance. The first step in plasmid transfer is contact between the donor and the recipient. The pilus of the donor cell recognizes and binds to specific receptor sites on the cell wall of the recipient cell. The plasmid then becomes mobilized for transfer when an enzyme cleaves one strand of the plasmid at a specific nucleotide sequence, called the origin of transfer. A single strand of the plasmid, beginning at the origin of transfer, enters the recipient cell. Once inside the recipient cell, a complementary strand to the single DNA strand is synthesized. When donor and recipient cells are mixed together, eventually all of the cells become donors.
Okay, transduction is another method of moving genetic material between bacterial cells. The vehicle for the movement is called transducing phage, which is essentially the bacterial equivalent of a virus. Basically, it's genetic material enclosed in a protein coat, which will then attack the bacterial cell. The phage DNA will be injected into the bacteria, and depending on the phage type, the phage can either integrate into the bacterial chromosome and remain silent for a period of time until such conditions as require its lytic production, i.e. the active generation of new virus particles, and in many cases the rupture of the bacterial cell take place. Um, or we can have phage that are purely lytic, that essentially infect their target cell, go into lytic production, and eventually the cell ruptures, and those viable phage go off and infect other cells. And you might well ask, okay, well, how does this move genetic material around between bacteria? Well, it turns out that some of these phage are a little sloppy in terms of the genetic material that they package into their virus particles once the lytic production of the phage begins. The result is that sequences of bacterial DNA can be captured by these virus particles, and then upon infection of novel cells, that genetic material can be introduced into another bacterial cell and obviously affect its genotype and its characteristics. So these are called transducing phages. So let's take a listen at how transduction works.
In generalized transduction, a segment of DNA is carried from one bacterial cell to another by a bacterial virus called a bacteriophage or phage. The phage attaches to the bacterial cell and injects its nucleic acid into the host cell. A phage enzyme is produced that breaks down the host DNA into smaller fragments. Phage DNA is replicated and phage coat proteins are produced. During formation of the mature phage particles, a few phage heads may surround fragments of bacterial DNA instead of phage DNA. The phage particle carrying the bacterial DNA infects another cell, transferring the bacterial DNA to the new cell. When the bacterial DNA is introduced into the new host cell, it can become integrated into the bacterial chromosome, thereby transferring genes to the recipient. This cell then multiplies and carries new genetic material.
Okay. Obviously, it's all well and good to work with bacteria as they're simple in the sense that they reproduce very quickly. They serve as effective DNA factories. It's easy to study their genetics because their genetics are simple compared to those of eukaryotic organisms. But at some point, what we want to do is investigate organisms that are a little bit more closely aligned with our own system, with the human system. And as a result, what we like to do is look at eukaryotic systems. Now, one system that I've kind of hopped over is yeast. Yeast is the simplest of the eukaryotes, or one of the simplest of the eukaryotes. It's a fungus, and it has all of the cellular components that you would expect to see in a typical eukaryotic cell. It has organelles, it has a nucleus, it has splicing, processing of RNA, and it has some novel characteristics such as the shift in yeast mating type that involve systems of site-specific cleavage and recombination of DNA involved in the mating process in yeast that have been harnessed in other systems, as we'll see in a bit, to generate transgenic animals. We'll talk a bit about yeast in class, but what I wanted to do is kind of hop over yeast for now and talk a bit about Drosophila melanogaster, the common fruit fly, as a system to study eukaryotic genetics. Essentially, this is an insect. Okay, it's an arthropod, and it has an extremely rapid life cycle, lasts about 12 days. It's inexpensive to maintain. It has a small genome, only 4 chromosomes. The chromosomes have been completely mapped and sequenced, so we know everything about the DNA of Drosophila front to back in terms of the A's, T's, G's, and C's, and we know quite a bit about the genes in Drosophila and what they do as a result of our ability to make deletions in these genes using a variety of techniques, and also as a result of the characterization of naturally occurring mutants in these genes. So all their traits are mapped with specified heritability patterns. You can see the life cycle here, relatively quick. Okay, we go from embryo to larva to finally pupa, and then once we finish pupating, we have the animal. Okay, so very, very quick. One of the interesting things about the development of the fruit fly is that during the larval stage, what we form essentially initially is a giant syncytia, but eventually we see the emergence of special structures called imaginal discs, which are locations of cells with predetermined fates for specific structures. They serve as valuable systems to study the effect of diffusible elements on gene expression and development. One of the ways that we control when and where certain structures appear in eukaryotic development is by the interaction of cells with molecules that are produced some distance away from that cell that will then attach to cell receptors and result in signaling cascades, which can then direct changes in gene expression in the nucleus. Okay, so very interesting phenomena whereby we can set up, in the case of the fruit fly, both an anterior-posterior axis and a dorsoventral axis through the result of these diffusible components. Okay, remember that the cell membrane has molecules embedded in it that serve essentially as antenna reporting on the extracellular environment, and not only that, often relaying information on changes in gene expression that are required to respond to those changes in environment in order for the cell either to find or assume its normal fate or to adapt to the environment around it. Okay, so these diffusible signals are critical. One of the things we also have to appreciate about the fruit fly is that there are a wide variety of mutations, both naturally occurring and induced, that allowed us in the early days of genetics to map the locations of genes relative to each other as a result of looking at recombinant phenotypes. In other words, you might have, let's say, a characteristic for eye color that would show up as different from the wild type. Wild type fruit flies have red eyes. You might end up, for instance, with a fruit fly that had orange eyes, and another fruit fly that had orange eyes, male and a female. And if you mated them and you observed that in the offspring, what you picked up primarily were wild type eye colors in the progeny, then you could make a reasonable assumption that those two mutations that produced that particular phenotype were on different genes for the pathway for eye color development in the insect. Whereas if they failed to complement, and you had progeny with all, let's say in this case, orange eyes, you could assume that the two mutations were in the same locus. Mutations in fruit fly characteristics pave the way as a result for the discovery that genes exist in allelic pairs, are located on chromosomes, and independently assort during meiosis. Further mutations demonstrated that the genes located on separate chromosomes segregate independently, whereas linked genes would fail to do so. Okay, so independent assortment and segregation were two of the hallmarks of Mendel's work, and the fruit fly allowed us now to give a physical location and an understanding for what Mendel saw as heritable units. We now, as a result of the fruit fly work, understood that genes lie on chromosomes, occur in pairs, and as a result, during meiosis, can independently assort and segregate. Okay? So an interesting model system. Now, how is it that we can modify this organism? Well, we can induce mutations in a variety of ways. Turns out that there's a very interesting genetic element in the fruit fly called the P element, which has the ability to move within the genome directed by an enzyme called transposase. As a result, these mobile genetic elements can excise themselves and then reinsert wherever there is an advantageous DNA sequence for them to integrate. Okay? So initially, Drosophila genetic analysis involved X-ray bombardment and assessment of the phenotypes that resulted from those mutations, and mapping was accomplished, as we indicated before, by comparing mutant recombinant strain phenotypes to parental phenotypes to assess the genetic and physical distance between the genes. As an example, let's go back to our eye color example. Let's say that you had one fly that had an unusual eye color, orange eye color, and then another fly that had vestigial wings and normal eye color. Vestigial wings just be very tiny wings. What you could do as a result of counting the progeny and assessing the phenotypes of the progeny is you could determine the genetic distance between those two mutations by looking at the number of recombinant progeny in the litter. Okay, the recombination distance is going to be directly proportional to the distance, the physical distance between the two genes. Okay, so if you have essentially ratios that would indicate that you only picked up now the parental genotypes, you could assume one of two things: either that the genes were on separate chromosomes, or that the genes were so far apart on the same chromosome that the recombination frequency would be sufficient to mimic as if they were actually on separate molecules. Whereas if you saw in the vestigial wing and the eye color virtually no recombination, okay, in the progeny, then you would assume that the distances between those two mutations were extremely short. Okay, so in our mating, recombinant phenotype would be flies that would have orange eyes and short wings. Okay, whereas the parental types would be orange eyes, normal wings, and red eyes and vestigial wings. Okay, so if we were to find large number of recombinant flies, okay, with both mutations in the same fly, then we could assume that the distances were either very great between those two genes or that they were on separate chromosomes. But if we failed to see any recombinant flies in the offspring, or very few, then we could assume that the genetic distance between the two mutations is quite short. Another phenomenon that occurs naturally in these flies as a result of these P elements that we were talking about earlier is something called hybrid dysgenesis, where what happens when you cross a P strain with an M strain in the egg, what will happen is that this will result in mobilization of these P elements, which are transposons. They can insert into viable genes during germ cell development, and this will disrupt the gene's reading frame or separate the structural gene from its control elements and produce a mutant. Frequent targets include genes required for germ cell formation. As a result, this explains the high frequency of sterility in dysgenic crosses. Okay, so this essentially is where pieces of DNA hop around. They move from place to place, and they can plop down in a variety of different locations, provided that that location has the proper DNA sequence for insertion. Not every stretch of DNA is going to have the right sequence of nucleotides for these P elements to insert. But what will happen when they do is that they can generate these novel mutants. And then you can do classic genetics by generating non-dysgenic crosses and you doing the classic complementation analysis and see whether or not these genes are tightly linked or whether they are in fact either on separate chromosomes or quite far apart on the same chromosome. Okay, so this dysgenesis is interesting. These transposable elements are interesting too because um, normally the wild type transposable element is simply some DNA sequences and the transposase enzyme contained between them. And so this mobile genetic element encodes a protein that's responsible for the movement of this piece of DNA. But we've found as a result of genetic engineering that we can insert into a plasmid containing the recognition sequences for the transposase a gene of our choosing. And then using a dysgenic cross, we can cause this novel piece of DNA to insert when and where we want. Okay, so the P elements can be used as vectors in the transformation of fly embryos. The gene of interest inserted into the modified P element, you can see here. Okay, there's our transposase gene on one plasmid, and there is our target DNA on the other. And then this can be excised from the plasmid and then can integrate wherever it finds an appropriate sequence in the fly DNA. And then this will allow us to study now the nature of the mutation that we've created. The interesting thing, of course, about this is that we can also, as a result of the DNA that we choose to put between these recognition sequences, sometimes do something very interesting, which is called promoter trapping, where we can put a structural gene in this transposable, this artificial transposable element that doesn't have a promoter next to it. So when it integrates near a control region for a particular gene, it does two things. Number one, it separates the wild type gene from its control elements. Number two, it serves as a cell signal tag that we can easily pick up in the fly. Okay, you can, for instance, include in these P elements proteins that glow under UV, or proteins that produce a color reaction in the presence of particular substrates. And the bottom line is that the mutant will display the characteristic gene expression in the tissues and at the times that the normal gene likely would be expressed. Now, that's not to say that always happens, but it happens a significant percentage of the time. Okay, so this is a unique and very interesting strategy. Now, if we go further, okay, this is sort of third generation recombinering, where we can hijack from yeast the flip-fart recombinase system, and we can produce now recombinations in areas that we desire by finding mutants that contain FRT sequences that we've crossed in to the chromosomes at specific regions, and then we can induce now a crossover where we want it, and this will generate a novel arrangement of genes in the insect. Okay, so this is a site-specific recombinase from yeast. The flip recombinase recognizes now the FRT sequence in the fly DNA, and then we can basically produce an artificial crossover really at any point in time, okay, but primarily during mitosis. As an example, we could take a let's say that we had a flip recombinase that was under the direction of a promoter that responded to changes in temperature. There are genes called heat shock genes that do just this. And we could induce the crossing over event by simply changing the temperature at a time and a place that we designate. And the result then would be a novel arrangement of genes whenever and wherever we want. In addition, as you probably have suspected, not only can we induce crossing over between chromosomes, but we can also, as a result of judicious use of these FRT sequences, generate deletions in particular genes when and where we want. And this has been applied not just in the fruit fly, but also in systems such as mouse and plant. So it's a pretty slick trick. Basically a tissue-specific promoter, which is inducible, right? We can turn the gene on when and where we want, and depending on the nature of the promoter, we can restrict it to a particular tissue type as well. So it all depends on the regulatory elements that you have access to. Okay, the next system we want to look at are plants, and this has a special place in my heart because I actually began my work in plants years and years ago, working with Nicotiana plumbaginifolia. This is a plant called Arabidopsis thaliana. It's again a rapid growing relative of the mustard, and it has again a completely sequenced genome, about 133 megabase pairs. It's an annual that has a 6-week life cycle. Okay, so very, very quick. It is a eukaryote. It is a plant, and you can see here how it is that fertilization is affected. We have the pollen grain generating a pollen tube, a fertilization tube that will fertilize the endosperm, but also, of course, the zygote. Okay, it's the endosperm that we actually treasure in terms of fruits and vegetables and so on. The zygote is actually the information that contains really all the other characteristics that are important in the plant. But the interesting thing, of course, about this is that because we know so much about Arabidopsis, we know that if we know if we've made certain changes in the DNA. Now we can do this in a variety of ways. We can introduce DNA exogenously through a device called a gene gun, which is essentially metal particles with DNA coated on the surface. We simply fire that into the plant tissue, and then a certain percentage of the cells will randomly integrate the exogenous DNA into their nuclear DNA. And if we've got exogenous DNA that expresses in a tissue that we designate with a promoter that we've chosen, then we have our transformed system. But of course, again, it's difficult using what they call bioballistics to actually direct your DNA to a site that you choose. Now we're going to see in a minute that random integration problem has become a thing of the past just within the past 5 years as a result of some novel recombinering techniques that we actually discovered in bacteria. But bioballistics and then this system, Agrobacterium tumefaciens, was the primary mechanism gene delivery in plants for years. Essentially the way Agrobacterium makes its living is that it carries a plasmid called the TI plasmid, which has the ability to integrate directly into plant DNA under the direction of its own promoter and its own polyadenylation signals. So you can see here the Agrobacterium. This, of course, the chromosome. This is the TI plasmid. The TI plasmid will basically produce now an integrant, okay, which can find a region in the DNA likely for integration. The foreign DNA will essentially pop in to the desired location, and then the benefit to the bacteria is that it directs the production of organic compounds that the bacteria feeds off of, and it generates a gall, okay, which is a sort of a plant tumor that houses the bacteria as they multiply. But what's of course interesting about the TI plasmid is again the virulence genes are similar in a sense to the transposon genes that we've seen in other systems. Essentially what they do is they will escort DNA that's between recognition sequences into the plant genome, and so you're not really restricted too much in terms of what it is you want to put in the plant. Standard practice these days is what we call bloom dipping, where we take a detergent and an agrobacterium culture, and then, of course, our DNA of interest engineered into a TI plasmid. We dip those blooms into the mixture, and then they'll eventually integrate into germline, and you can produce transgenic seed. But again, you can't, in this case, necessarily determine where the DNA is going to go. It might go into a region of DNA that is amenable to gene expression. Then again, it might integrate into areas of DNA we call heterochromatic, where gene expression is dampened for some reasons that we understand and some really that we don't. But what I wanted to show you here was just a look at the mechanism of agrobacterium integration.
This video is an illustration of the mechanism of agrobacterium using the genes for herbicide and antibiotic resistance. The genes are first inserted into the TI plasmid between the left and right borders. Since agrobacterium-mediated transformation is a binary system, the TI plasmid must then be inserted into agrobacterium using electroporation, sonication, heat shock, or some other method. Next, explants must be inoculated with agrobacterium. In this case, a scalpel is used to pick a colony from the plate and then wound the leaf of a plant. Agrobacterium then attaches to the plant's cell wall and transfers the tDNA portion of the TI plasmid to the host. This tDNA portion, which contain the genes encoding herbicide and antibiotic resistance, are then randomly inserted into the host's genome. The explants can be further cultured under sterile conditions on agar plates to form a callus, which is an undifferentiated mass of cells. The transformed callus can subsequently be cultured on agar plates containing the appropriate plant hormones and antibiotic to select only those plants that have been transformed. The result are whole transgenic plants exhibiting the trait of interest, in this case, herbicide resistance.
Okay, our last system, and the one probably closest to the human system, is the house mouse Bus musculus. Okay, mouse development parallels human development on an accelerated time scale. After fertilization, early cell division occurs every 12 to 24 hours. This is again internal fertilization, just like humans have. Formation of the morula seen at the 16 cell stage. Cells in the outer region don't contribute to the embryo, but they become the placenta, and the remaining cells form the inner cell mass, which will eventually be the mouse. At the 64 cell stage, there's 13 inner cell mass cells, which will form the adult mouse tissues. The blastocyst is now ready for implantation in the uterus, at which point the placenta will develop. Once the placenta is established, the embryo enters gastrulation, followed by the establishment of the hypoblast and the epiblast, which will form the ecto and endoderm. After the formation of these two germ layers, a third germ layer, the mesoderm, will be produced as cells migrate through a dorsal primitive streak that runs essentially down the back midline of the disc, which will eventually be the mouse. And this will result in the establishment of all three germ layers, the ectoderm, the mesoderm, and the endoderm. And the reason that that's a big deal in development is because those three germ layers are responsible essentially for producing all the other adult structures. The ectoderm producing the skin and many components of the nervous system, the mesoderm producing the cardiopulmonary musculoskeletal system, and the endoderm producing the lining of many of the internal organs. And that's a bit of an oversimplification, but that's basically it. Fetal development then follows this, then followed, of course, by neonatal development. Interesting thing is that embryos can be rescued from the reproductive tract of a female mouse and microinjected with DNA constructs containing a transgene that you pick out. The transgenic strains can be characterized in a variety of ways. The most popular way is polymerase chain reaction, which we're not going to do in this course, but you guys will do in 205. Basically, it's a method of amplifying DNA using artificial pieces of DNA called oligonucleotide primers in the presence of a thermostable polymerase and a thermocycler. Also, the building blocks of DNA are required, and what happens is that the tube is alternately heated and cooled. The result is that as a result of the cycling temperatures, you generate many copies of a particular region of DNA, which you can then analyze using gel electrophoresis, or you can actually sequence and look at the arrangement of nucleotides in that particular piece of DNA. Okay, so this is one of the things that replaced southern blotting relatively early on in its development, because it's much less expensive and it's much more rapid. So we can characterize these strains, and then germline transmission will be demonstrated if we pick up the same genetic modification in the progeny. That means that you have managed to get your transgene to germline. Okay, that it's not always a guarantee, right? That if you inject a developing embryo with DNA of your choosing, number one, that it'll integrate, and number two, that once it integrates, that the cells it integrates in will contribute to the formation of either sperm or eggs. And if they don't, then you really haven't affected a change that you can study extensively, because you're not going to be able to look at progeny. Okay, so the key here is to get what we call germline integration. Another way that we can deal with manipulating the genome in the mouse is through the use of embryonic stem cells that come from the early intercell mass. They can be cultured outside the mouse and induced to form any adult cell type in the presence of the appropriate growth factors. These ES cells can be used to execute targeted recombination with positive-negative selection, and this is just one example of positive-negative selection. What you're looking at here is a construct that contains the neomycin gene and the thymidine kinase gene, and the idea here is that you'll get many integrations as a result of your introduction of your target DNA. And again, how do you get the target DNA in there? You can inject it, you can electroporate it, use electricity essentially to open the membrane up and get the DNA in there. You can deliver the DNA with special positively charged lipids that'll fuse with the cell and throw the DNA in. The choice of delivery method is going to depend on really the characteristics of your cell type. For cell types, certain methods are much better than others. It's hard to say why in many cases, but again, we have a multitude of potential methods to get exogenous DNA inside this or any other eukaryotic cell. But once this happens, okay, in the case of our positive-negative selection here, our construct contains neomycin, and so you grow these transformed cells on culture media containing neomycin. Cells that don't have the neomycin gene are going to die off, and then the cells that have the neomycin gene will survive. But that alone doesn't tell you that you've got an integration in your construct. Okay, it just tells you you've got an integration, period. You want to find out whether you've got an integration in your construct by checking for the absence of the thymidine kinase gene. In other words, you want the recombination to essentially eliminate the TK gene and to conclude only the neo-interrupted portion of the structural gene that you've designated. Okay, and you can see here the difference between a homologous and a non-homologous recombination event. If you get that integration, okay, where we are neo-resistant but TK sensitive, then in all likelihood you've got the integration targeted the way you want. The problem is, at least in the old days, that this was often a difficult task, okay, because you had to design vectors with extensive homology to your target. You would engineer your neo or whatever cassette you wanted in the middle of your gene, and you would produce either a recessive mutant or a conditional mutant, depending on what you put in the cassette. What happened often, though, is that you would get an integration totally outside your designated locus, or you might get an integration, a partial integration, and you would lose the rest of the locus in the integration event. Okay, there's all kinds of ways that this recombination can go that can result in products that aren't useful for your research. And often this was a very expensive endeavor and very time consuming. So if you had an error in the construct, or if you had a problem with the integration and you couldn't make your mouse, then you were pretty much out of luck as far as characterizing your gene goes. But with the emergence now of a new technology called CRISPR, this problem of site-specific integration has to a large extent been abrogated. This has reduced the cost of generating transgenic mice many-fold. Okay, so what I want you to do here is listen to the CRISPR approach for generating not just transgenic mice, but really any sort of transgenic organism using these now patented techniques. Okay, so that covers what we wanted to talk about in terms of genetically modified organisms. What I want to do now is talk a little bit about what you guys are going to be doing in the next 2 weeks, which is transforming bacterial cells and then isolating DNA out of those cells and characterizing it by running it on a gel after you cut it with restriction enzymes. So we have to think again about the physical properties of DNA, and what we remember about that is that it has a specific temperature at which it's happy, it has a specific pH at which it's happy, and it has a specific salt concentration at which it's happy. In addition to that, we want to eliminate cofactors for enzymes that would degrade the product. So one of the ways that we deal with that is that in many solutions we work with in DNA, we use an organic buffer called tris, and the chemical structure of tris is given here. Its useful buffer range is between 7 and 9, and this coincides with physiological pH, which is good for us because most DNA is active at physiological pH. It's typical of most living organisms. This and the fact that it's low cost make it one of the most common buffers in the biochem laboratory. It's also used as a primary standard for acid solutions for chemical analysis because it's so stable at holding the pH at temperatures you're going to work at. Okay, so this is what tris looks like. One of the things you're going to do is you're going to make a tris solution at a specific pH, and that pH is 8, and the recipe is given here. Okay, so you can see the amount of tris you need, the amount of water you need, and then you're going to pH it using hydrochloric acid. That's concentrated hydrochloric acid, and you're going to add it dropwise to the stirring solution until the pH meter reads 8. And I'll show you guys how to do this. This is just essentially a thumbnail sketch of how you're going to make these buffers. Once you've got the proper pH, you can add water up to the desired volume, okay, 100 mils in this case, and then you can autoclave it and store it at room temperature indefinitely. Okay, and you're going to use that to make a wide variety of solutions. Another important organic component in a lot of DNA solutions is ethylene diamine tetraacetic acid, also known as EDTA. Structure is shown here. It's widely used for scavenging metal ions in biochem and molecular biology, particularly magnesium, which is a cofactor for many nucleases. The depletion of this ion renders the solution stable with respect to DNA structure. So it deactivates metal-dependent enzymes either as an assay for their reactivity or to suppress DNA damage, which is what we're going to be using EDTA for. And to make EDTA pH 8, this is the recipe. Okay, you pH this with sodium hydroxide pellets. Now you want to be very careful with sodium hydroxide because it's caustic. It'll burn your skin. It'll actually react with your skin, forming a slippery solution in a reaction called saponification. It's literally dissolving your skin into soap. So when you work with sodium hydroxide pellets, what you want to do is weigh out the requisite amount, okay, or add spoonfuls of the pellet until you've reached your appropriate pH. You don't want to leave the lid off of the sodium hydroxide because it's a hygroscopic reagent, meaning it sucks water out of the air, and that's going to essentially reduce its efficacy. In addition, it'll cause you to get big, huge chunks of sodium hydroxide inside the bottle that are difficult to bust apart and use for future applications. Once you've reached your appropriate pH, what will happen is the EDTA will go into solution. You'll know that because the solution will become clear. And then you add water to volume, and then you can autoclave it and store at room temperature indefinitely. Now, one of the things we have to understand about making solutions is that you always want to start by putting water in a beaker, okay, and get that stirring first. You don't want to throw your powder in there and throw your water on top of it, because what will happen is you'll just make a big insoluble crust that'll be very difficult to get to go into solution. So you want to start stirring about half as much water as you predict you're going to need, okay, with a stir bar on the stir plate, and then you add the powder carefully, okay, to the stirring mixture, and then you do the pHing. Okay, pHing follows a protocol. I'll show you how to standardize a pH meter using buffers. Okay, and then again, once you've got this stuff made, it's going to last you for a long time. Okay, we're going to use it in a variety of buffers in gel electrophoresis and in DNA isolation. And then finally, you want to make some good old 6 molar sodium hydroxide in order to pH the other stuff. 6 molar sodium hydroxide, you want to make it always in a plastic container because it etches glass. Use about 24 grams of sodium hydroxide pellets. Add distilled water to 100 milliliters. Do not autoclave this. Okay, it is sterile as is. Nothing can live in 6 molar sodium hydroxide that I know of, and you can store it at room temperature indefinitely. One of the things you'll notice about the sodium hydroxide solution is going to get very hot when you make it. That's natural. It's an exothermic reaction, so don't be worried about the generation of steam as you stir this into the liquid. It's natural. And again, as always, with this and all your other solutions, you want to gown up, you want to glove up, you want to put on your protective eyewear, no open-toed shoes, and so on. Okay. In addition, every reagent that you make, you want to label what it is, you want to label the date you made it, you want to put your initials on it. Okay, and the reason for that is A, so you know what it is, B, so you know when it was made, whether or not it could go bad, okay, and also so you know who it belongs to. And also, as you use these reagents, you want to include in your notebook, obviously, the lot number, the manufacturer, and the catalog number for the reagents that you use. 5 molar sodium chloride, important reagent. Pretty simple here. About 29 grams of sodium chloride added to 50 milliliters of stirring water. Let it dissolve, make it up to 100 milliliter volume. You want to autoclave this. You can store it at room temperature indefinitely. 3 molar sodium acetate at 5.2 is important because we're going to use this as a method of precipitating DNA, which I'll tell you about in a minute. Essentially, about 24.5 grams of sodium acetate. Add 50 milliliters of stirring distilled water. Let it dissolve. pH it to 5.2 with glacial acetic acid. It's going to smell like very strong cider vinegar. Again, glacial acetic acid is a reasonably strong acid, so you, again, want to make sure you've got your gloves and your glasses and your lab coat on, your PPE on. And then once you get to 5.2, you can stop. You can add water to 100 mils, autoclave it, and store it at room temperature indefinitely. This should last you centuries. One of the things that we're going to make that you're going to use to dissolve DNA in stably is TE. Okay, TE is a buffer in which DNA will remain stable at room temperature almost indefinitely. pH 8 is the pH that DNA likes to be at. So TE pH 10 is pH 8 rather is a standard buffer for dissolving DNA, and it maintains a stable pH and chelates the metal cation so the nucleases don't tear the DNA up. The subscripts in TE refer to the millimolar concentrations of each reagent. So T 10 millimolar, E 1 millimolar. Okay. So the way that you generate this, okay, is you use your stock solutions that we made the previous inserts. 20 microliters of sterile half molar EDTA at 8, 100 microliters of sterile 1 molar tris at 8, add 9.88 milliliters of sterile water, mix it, and there you go, you're done. Okay, you got enough TE to last you a lifetime. Sometimes when you manipulate DNA, you're going to have to change the volume that the DNA is in. In order to do that, what you have to do is precipitate the DNA and redissolve it in another solution that fits the buffer conditions you need for that process. So ethanol precipitation is essentially a way of stealing water molecules away from the DNA molecule and causing it to form a solid, which you can then spin down to the bottom of the tube, dry off, and then redissolve in a buffer of your choosing. Okay? So what happens when we ethanol precipitate DNA is first we add salt to the buffer. The salt steals the water from the DNA becomes less soluble, and then the alcohol finishes the job, okay, of dehydrating the DNA, and you form a solid mass. It'll come out of solution. You chill it generally between 15 and 20 minutes at -20 to help the precipitation go along. Again, remember that solubility decreases as temperature decreases, and then you're going to spin that tube down 15 minutes max speed in a microfuge, and you'll generate a white pellet at the bottom of the tube. You'll then pour the liquid off the top of the pellet. That's called the supernatant. That doesn't contain anything you want to keep. And then you'll wash that pellet one time in 70% ethanol to remove any salt, and then let that dry, and then you'll dissolve it in the buffer of your choosing. Could be TE, could be any number of things. Okay, so that's the protocol right there. Sodium acetate 3 molars added at 1/10 volume. If we have glycogen around, it's not a bad idea to throw a little glycogen in there. It helps to pull the DNA out of solution more effectively, and it gives you a pellet that you can see. But we don't always have glycogen around, so that's a totally optional thing you can do. You add 2 volumes of cold ethanol, you mix, you precipitate at -20 for between 30 minutes and an hour. And then you centrifuge for 15 to 20 minutes at maximum speed in a microfuge, max speed between 14 and 16,000 G. Okay, you'll see a visible pellet. You'll pour the supernatant off. You'll add a volume of 70% ethanol and water, and then you'll centrifuge for 5 minutes at max speed in a microfuge. Pour off the supernatant. Be careful here because now the pellet is loose and could dislodge from the bottom of the tube. Let the supernatant drain into a Kim wipe, and then resuspend the sample in your desired buffer. And then next week, what we're going to do with your isolated DNA, you're going to, after you've transformed E. coli, you're going to isolate DNA from it, and then what we're going to do is we're going to run it, cut it, and run it on a gel. Okay, so gel electrophoresis is just a method to analyze a DNA molecule for size. Basically, it separates the DNA molecule according to the length of the DNA. The slower DNA fragments are the bigger ones, and they stay near the top of the gel, near where you load at the well, and the smaller DNA fragments run more rapidly towards the positively charged cathode, and that affects now a size separation. Okay, the matrix that we're going to use is something called agarose, which is derived from seaweed. And it provides a meshwork through which the DNA must work itself, and that's why big molecules don't move as fast as tiny ones, because big molecules have a harder time getting through the matrix than the small ones, and so that affects a separation according to size. And then we can visualize the DNA. We're going to use something called cyber green, which chelates the DNA and glows green under UV. We'll be able to see our bands, and we'll take a picture of it. We also want to make running buffer in order to perform our gel electrophoresis. We use a 50x stock. 50x just means that the stock is 50 times more concentrated than the working concentration. So what you do to make 1 liter of 1x TAE, 20 mils of 50x, 980 mils of water, mix it, store it at room temperature indefinitely. Okay. And then we've got loading dye in the lab, but I wanted to show you this in case you want to make your own. We have all the reagents for this. The nice thing about the loading dye is it sinks the DNA into the bottom of the well, and the dyes themselves separate according to relative size. Okay, the xylene cyanol and the bromophenol blue mark the midway point and the terminal point for most of the DNA runs you're going to do. The xylene cyanol looks kind of aquamarine, and the bromophenol blue looks blue. And again, you can use that essentially as a way to determine how far your gel has run and whether it's time for you to stop the current and take a picture. Okay, and of course, in order to run a gel, you have to make a gel. So this is the recipe for 0.8% TAE agarose, 100 milliliters of 1x TAE, 0.8 grams of agarose. Swirl it, wave it until it's dissolved. You'll know because it'll go clear. Watch it while you wave it because you don't want it to boil over. Okay, but it'll start to go clear. It's a little bit like making Jello. Okay, if you've ever made Jello before. Once you're dissolved, then you're ready to go ahead and pour your gel in the gel casting tray with a comb of the appropriate well number and depth, and then we can go ahead and load your samples. In the event you want to separate something that's much tinier, you might need a higher percentage agarose gel. This is the recipe for 2%. Okay, 100 mils of 1x TAE, 2 grams agarose. Shake it, wave it just like before. Watch it so it doesn't boil over. And then of course you can store both of these as long as you want. Once you wave the agarose, get it to go into solution. When it cools again, what will happen is it'll turn kind of a pale gray, and of course it'll become a semi-solid. Okay, of the consistency approximately of Jello. Okay, so what's on tap for this week is we're going to transform chemically competent E. coli with a plasmid that I call plasmid X. Okay, I'll order these cells. The thing about ordering these cells is they'll come in when they come in, and then we'll use them that day or the next day. Okay, they don't keep because in order for them to keep, we need a minus 80-degree fridge, and we don't have one of those. We have a minus 20. So these kind of need to be ordered fresh, and then we just go for it. But anyway, first thing you're going to do is make some. We've got LB plates in the fridge, but you're going to turn those LB plates into LB AMP XGAL IPTG plates. And you might well ask, well, what's this other stuff, AMP and XGAL and all that? Ampicillin is just a drug that will select for bacterial cells that have taken up plasmid that contains the ampicillin resistance gene. XGAL is a substrate that when cleaved by beta-galactosidase produces a blue color. And IPTG is a molecule that's called a gratuitous inducer, and what it does is it locks the beta-galactosidase gene in the on position and causes it to continually transcribe in the bacteria. So you're going to add these components to your plates. The reason that I have you guys make regular plates and turn them into selectable plates is because the XGAL and the IPTG are kind of pricey, so I don't want to waste them, and this is the best way to use them without wasting them. You just sort of spread the stuff on there, okay, as shown here with a hockey stick, and then you let that sit in, okay, let it sink into the agar for about 20 minutes, and then your plate is ready. Okay, so once you've spread your plates and they're ready to go, then they'll keep for about a week. Okay, and that'll be fine for the window we're going to do our transformations for. You're going to do a transformation using a plasmid that contains an ampicillin resistant gene. And you also do a transformation with sterile water as a negative control. And then you're going to incubate after that transformation at 37 degrees overnight. You're going to transform electrocompetent E. coli with plasmid X and plate that on LB and incubate that 37 degrees overnight as well. Okay, so we've got a negative control and we've got a transformation control. And the protocol is here. I won't go through the whole thing because you'll see that in the handout. But it's pretty straightforward. You just get the chemically competent cells on ice. Once they thaw, you go ahead and you add your water in the case of your negative control competent cells, or your plasmid X in the case of your actual experimental sample, to another aliquot of the competent cells, and then you let them sit on ice for 30 minutes, and then you heat shock them at 42 for 30 minutes. We'll do that in the water bath, and you put them back on ice for 5 minutes, and then you're going to add a milliliter of LB, okay, like the LB you've been using, the non-selective media here to give the plasmid, if it's in there, a chance to express. And then you're going to take that 1 mil shaking culture and you're going to plate it, okay? For your DNA transformation, you're going to do a straight plate. You're going to do a 1 to 10 and a 1 to 100 plate. So for each of you, you're going to need 3 plates for your experimental, and you're going to need 1 plate for your negative control. So minimum of 4 IPTG XGAL amp plates, LB amp plates for each of you to do this experiment. Okay, so I imagine that'll probably be what we'll do first. We'll get your plates ready, and then we'll do your transformations, and then next week we'll isolate your DNA and we'll cut it and run it on a gel. Okay, all right. This takes care of the Week 8 podcast. Again, if you have any questions, let me know, and I'll see you guys in class. Thanks.